Enzymes means what? Nil Jama. No. Enzymes are biocatalysts, about biocatalysts, ekdom correct. Up to biocatalysts, it is correct. Very good. Biocatalysts, তাই তো? তাহলে enzymes are biocatalysts. Let's see what I have written. Correct. You are correct. ভুলে গেছিলাম, দেখে নিলাম। Enzymes are biocatalysts. Catalyst মানে? Wow, you can read and tell, right? Very good. That means you can read English. This proves you can read English, right? So catalysts are something which participate in the reaction. It neither initiate nor terminate the reaction, but rather it just increases the rate of the reaction. Okay, in both reactions. Enzymes, unlike organic or inorganic catalysts, enzyme itself is an organic substance, they are heat labile. Why? Because most of the enzymes are protein in nature and few are there which are non-protein in nature. Ribozyme, very good. Give one example. Give one example of ribozyme. To synthesize protein, what do you need? No, not as a building material. What is that? Peptidyl transferase. Right? That is a ribozyme. That is a ribozyme. So evolutionarily it was chosen. Why? Because if you require RNA for RNA synthesis, then who would have discovered the first RNA? If you require protein for protein synthesis, then who made the first protein? This question will come now. Before egg came before or hen? Do you know the answer? Answer is there. Now the answer is there. Okay, fine. So for synthesizing protein, we have RNA or ribozymes. Okay, example is peptidyl transferase. That is nothing but 28S rRNA by nature. Anyway, now being a protein there, heat labile. Heat labile means if you give a heat, then it will be destroyed. Suppose you want to kill a person and you give a cocktail of snake venom in hot water. Will the person die for drinking? No. If you give in the cold water? No. Snake venom must get mixed with the blood unless the person is having gastric ulcer or wound in the GI tract. Hardly anyone is there like that. So there is a possibility that he might die. But there is no. Theoretically, he should not die. Specific for substrate. Now this is something like, say glucokinase, hexokinase. Hexokinase is specific for hexosugar. Glucose is specific for glucose. So each enzyme has its own specific substrate. Right? This is very important criteria of enzyme compared to catalyst. Low activation energy. By virtue of nature or the theory of perpetuality, whatever you say, if I keep a glucose and an ATP side by side, then there is a possibility after a few hundred years this glucose will be converted to glucose 6-phosphate without any enzyme. But if it happens so, then our lifespan should have been thousands of years longer. We would not have grown so faster. But that never happens. That means someone plays some role. What is that? That enzyme lowers the activation energy of the reaction so that the reaction can take place very fast, correct? Activity may be regulated. You are very hungry, correct? You are eating, then you stop eating. Why? Because you are not hungry anymore. If you are not having food inside the stomach, will there be any enzyme for digestion? No. If a food is there, enzyme will be there, correct? So, we can regulate the production. How? By hormones. When you are feeling hungry, all secretions will increase whenever you see food. Suppose you are a biryani lover. If you see biryani, what will happen? Automatically you will start salivation. But suppose you are a shukto lover. Shukto? Heard of shukto? A vegetarian dish. Will you salivate the same way as you salivate with the shukto in case of biryani? No. Correct? So, these are the things. So, we can regulate the enzymes, secretion, synthesis, etc. and its destruction also. Besides this, there are some similarity with catalysts. They are the following points. Just go through. Nothing new here. Both are needed in minute quantities. Small quantities required. They cannot initiate reaction or terminate the reaction. They remain unchanged after reaction. This is somewhat questionable. In some reactions, the physical form of the catalyst might be changed, but not the chemical form. Decreases activation energy. I will come to this very interesting subject. Combine with substrate during reaction. That means what? What is this? Proximity. A proper proximity is required. We cannot increase our population when husband and wife are miles apart. Can we? Do you have any doubt? No doubt? So, without proximity, no reaction, no product. Reaction catalyzes are reversible. By the laws of nature, it is said all reactions are reversible, depending on certain conditions. What is the condition? If we continuously remove one of the product from the reaction vessel, then the reaction will move towards that direction, right? If we change the temperature, some reactions are there at lower temperature, it will react from left to right, at higher temperature, it will react right to left. Same happens with pH, lower pH, left to right, higher pH, right to left. So, all these are decisive factors for enzymatic reactions or catalytic reactions. Now, what are the dissimilarity or differences? These points are important. First one, milder reaction conditions. If you can recall, what was the reaction condition for Haber's process for production of ammonia? নীলজামা তুই আগে উত্তরটা ভালো দিয়েছিস বলে জিজ্ঞেস করলাম, রাগ করিস নি? হেবার্স প্রসেসটা মনে আছে? হ্যাঁ, অ্যামোনিয়া তৈরি করে। হ্যাঁ, ওটা বলে কন্ডিশনগুলো বলতে পারবি? 2 অ্যাটমসফিয়ারিক প্রেশার, টেম্পারেচার কত ছিল? 370 ডিগ্রি সেলসিয়াস, তাই না? ছিল না? সো ভেরি এক্সট্রিম কন্ডিশন, হিট, high atmospheric pressure, all these are required for a chemical reaction to take place. বাংলা বুঝতে পারছিস তো? গুড। ইজ দ্যার এনিওয়ান, আচ্ছা এখানে কেউ এমন আছো যে ইংলিশ বুঝতে পারো না, একেবারে কষ্ট হয় ইংলিশ? ইংলিশ ইংলিশ বাংলা? How many of you don't know Bangla? Oh, only 4. Then we can take class in Bangla. as per NMC rules, 70% of the class can be taken in the local language. Are we following the rule? Where? Very good. From now on, I will follow. Let's see. Right? Anyway, sit down. বস বস তা কি বলছিলাম আমি Haber's process. So the chemical reactions require extreme conditions, high temperature or very low temperature or high atmospheric pressure, but our body is accustomed and function well at 1 atmospheric pressure and temperature 37 degrees Celsius, our body temperature, pH 7.4, nothing is very extreme, correct? So that is the beauty of enzymatic reaction, that it can convert the substrate to product at milder reaction conditions, correct? Higher speed. When I say higher speed, means enzymatic reaction produces higher speed of the reaction. How high? The slowest reaction is at least 1 lakh times, means 10 to the power 6 times faster than the fastest chemical reaction. A million times faster than any chemical reaction, any enzymatic reaction. That is why a fetus can grow in such a huge rate inside the mother's womb. After 1 week, it is just like a nail of a thumb. After 3 weeks, a palm, this much length. And after 15 years, see? Sikira, right? Almost 15 years. After 15 years, I stopped. I was not the tallest among the class. High specificity. In many chemical reaction, the byproducts sometimes becomes more valuable. Like in an acid factory, the major byproduct is sodium chloride, which is being sold in packaged salt for our consumption. Natural salts and this chemically produced byproduct. Which one is cheaper? Chemically produced byproduct, because they anyway they have to dump it somewhere else. Why not inside our stomach? So byproducts means, more byproducts means wastage of substrate, wastage of resources, wastage of energy, money and time. But enzyme reactions are highly specific. Hardly any byproduct is formed. With glucose plus ATP, glucose 6-phosphate, that's all. Scope for regulation. When I'm eating a lot, I'm gaining weight. When I'm starving, the fat synthesis declines. This nice structure will disappear if I don't eat for 7 days, 15 days, 30 days, or if I go to jail, want to lose weight, just eat the food in jail, Indian jails, you lose weight, except few. There must be some other arrangements inside the jail. So if you go like a normal convict, you lose weight. Right? Scope for regulation is there. But once the chemical reaction started, we cannot stop it. It will finish, just like an atom bomb. Just like an atom bomb. Once it is ignited, then it will blow it up. Then you cannot stop. Now we come across this part. What are different parts of an enzyme? Enzymes, mostly being protein, Say out of 100 enzymes, 90 enzymes are proteins. Okay, rest all are some other substances. But these terms we come across very often, Apoenzyme and Holoenzyme. Holoenzyme means not all enzymes, these terms are not true for all enzymes, but few enzymes where in addition to protein part, some non-protein part is also present in the enzyme. Correct? The non-protein part can be a structural component of the enzyme, it can be a functional component for the enzyme. So the protein part of the enzyme is called Apoenzyme. Ki bole? Apoenzyme. So the Apoenzyme is a protein part. Holoenzyme, the Apoenzyme plus the non-protein part. Now this term, so the Apoenzyme plus something makes Holoenzyme. What is that? Apoenzyme plus something makes Holoenzyme. What is that? Huh? Cofactor, not coenzyme. Apoenzyme plus cofactor forms Holoenzyme. Cofactor is a non-protein part. So cofactor can be of two types, organic and inorganic. The organic cofactor, which is loosely bound, number one. Organic, number two. Loosely bound, number three. Dialyzable means can be separated by dialysis. If the cofactor has these three properties, then the cofactor is called coenzyme. Ki bolam? Organic in nature, loosely bound, dialyzable means can be separated by dialysis. Then it is called coenzyme. The other things are called, the rest of the part of coenzyme, cofactor is called cofactor. Among them, it can be metal or non-metal. Correct? If it is a metal part, then again two things will come. Whether the metal part component is a structural component or functional component. Suppose a zinc in carbonic anhydrase, it is functional component. Magnesium in hexokinase, it's a functional component. Like that. So iron in heme, it's a structural component, without which the structure will not form. Right? So they are called either metalloenzymes or metal activated enzyme. Either metalloenzyme, where the metal ion is the structural component, or metal activated enzyme, where the metal part participate or help the enzyme to act. Now this particular table is very, very important. Why? Because this table has got all the names of vitamins and their corresponding coenzyme forms. Mostly water-soluble vitamins participate as coenzyme, mostly. Correct? Fat-soluble, few fat-soluble vitamins also have coenzyme activity. We will come to that later. For the timing, we will try to remember the water-soluble one. The first one here, it is biotin. Biotin participates in any reaction where carbon dioxide is added. Means an enzyme whose name ends with carboxylase. Can you remember any enzyme with carboxylase? If you cannot, don't worry, you should not, it is not a part of your acetyl carboxylase, malonyl carboxylase. So what is it doing? They are adding one carbon dioxide. So these enzymes require biotin. What is the use of biotin? Bolo? Biotin, other name is vitamin B7 vitamin hair, vitamin H. কত অ্যাড দেয় টিভিতে है ना, बायोटिन माखুন, চুল গজান, है ना. So vitamin H, biotin is used for vitamin H, for hair growth, hair health. Coenzyme A. Coenzyme A. Coenzyme A is a coenzyme form. The vitamin form is pantothenic acid. Pantothenic acid, pantothenic acid. There is a protein called 4-phosphopantothene, is used in synthesis of fatty acid, which has pantothenic acid as a part. What is the function of coenzyme A? We find coenzyme A in these compounds. Let me tell you first the compound, then try to guess what is the function of coenzyme A. Acetyl coenzyme A, malonyl coenzyme A, acyl coenzyme A. So, what is the function of coenzyme A? To carrier of acyl group. Acetyl group. Acyl group means what? CH3, CH2 dot. Right? It's called acyl. If CH3, CH2 হয় তাহলে acetyl, মিথাইল, ইথাইল, है ना, acyl group হয়ে গেল। তাহলে এরকম যদি অ্যালিফ্যাটিক গ্রুপ হয়, চেইন হয়, তাকে যে ক্যারি করে সেটা হচ্ছে কোএনজাইম এ। নেক্সট হচ্ছে বলো এটা কোন ভিটামিনের পার্ট? ভিটামিন বি টুয়েলভে অনেকগুলো ফর্ম আছে, তার মধ্যে একটা ফর্ম হচ্ছে 5' অ্যাডেনোসিল কোবালামিন। 5' অ্যাডেনোসিল কোবালামিন যেটা থাকে এটাকে বলা হয় কোএনজাইম অ্যাক্টিভ ফর্ম। ঠিক আছে, আদার ফর্মগুলো যেমন কি কি ফর্ম আছে, যেমন সায়ানোকোবালামিন, আর কি ফর্ম আছে মিথাইল কোবালামিন, আর কি ফর্ম আছে হাইড্রক্সি কোবালামিন। কোনটার কি ইউজ? 5' অ্যাডেনোসিল কোবালামিন হচ্ছে কোএনজাইম অ্যাক্টিভিটি, মিথাইল কোবালামিন স্টোরেজ ফর্ম, কার মধ্যে? অ্যানিমালস। সায়ানোকোবালামিন স্টোরেজ ফর্ম, কার মধ্যে? প্লান্টস। হাইড্রক্সি কোবালামিন the injectable form, যেটা আমরা ইনজেকশন দিই, ইনজেক্টেবল ওয়াটার সলিউবল ফর্ম, সেটা হচ্ছে হাইড্রক্সি কোবালামিন। সেরকম একটা ফর্ম হচ্ছে 5' ডিঅক্সি অ্যাডিনোসিল আর 5' অ্যাডিনোসিল কোবালামিন। ঠিক আছে, তাহলে ভিটামিন বি টুয়েলভ একটা পার্ট। এফএমএন এর পাশাপাশি আরেকটা আছে এফএডি। এফএমএন মানে ফ্ল্যাভিন মনোনিউক্লিওটাইড, এফএডি মানে ফ্ল্যাভিন অ্যাডেনিন ডাইনিউক্লিওটাইড। এই দুটোই কোত্থেকে ডিরাইভ হয়? দিস টু আর ডিরাইভ ফ্রম ভিটামিন B2 or রাইবোফ্লাভিন, রিবোফ্লাভিন, যা বলো। আচ্ছা, ভিটামিন না ভাইটামিন? হাউ মেনি অফ ইউ থিঙ্ক ইট ইজ ভিটামিন? হাত তোলো। গল্পটা সুন্দর গল্প আছে। গল্পটা হচ্ছে, the story behind this is when it was first discovered, they discovered only a part of the vitamin which contain amino group. So they named it is vital for life which contain amino group, so vital amine. Later on they found there are many compounds in this group which don't have any amino group, like just like vitamin A, vitamin D, vitamin E, they don't have any amino group, but still they are vitamins. So then they removed E. To change it, they started vitamin. And then some people, like the driving school, they changed. England different, America different. Right hand side driving, left hand side driving. Lipoic acid. Lipoic acid. What is a vitamin? The coenzyme form is actually lipoic acid or lipoamide. Lipoamide, just লিখে রাখো এটা। Lipoamide. The vitamin name is কি হবে বলো? Anyone? তুমি বলো, বলো। Name the vitamin responsible for this one. দেখি Who got highest marks in NEET biology? কে পেয়েছে হাইয়েস্ট মার্কস? তুই পেয়েছিস বায়োলজিতে? কত পেয়েছিস? বায়োলজিতে কত পেয়েছিস? বায়োলজিতে, নিটের বায়োলজিতে কত পেয়েছিস? আর একটু জোরে বল। 29 পেয়েছিস? ভাগ করে নম্বর দেয় না নিটে? কেমিস্ট্রিতে এত ফিজিক্স দেয় না? হ্যাঁ, ও বস বল, কে পারবে? So apart from the vitamins you know by far, there are few other molecules which are called minor vitamins. One of them is lipoic acid. One of them lipoic acid. And the next one is, other one is choline. Right? So these are all minor vitamins. Sit down. See, so many things. It was there in your syllabus, +2. I'll ask you only +2 only. This is what? NAD. What is the difference between NAD and NADP? You know, two forms NADP. In plant, NAD mostly in animals, NADP for synthesis, NAD for other than synthesis. Niacin or nicotinic acid. This nicotinic acid has nothing to do with nicotine, but yes, if you can synthesize and impregnate the pathway, the enzymes responsible for pathway to convert nicotine to nicotinic acid, then a Nobel Prize is waiting for you. All cigarette companies will give you money. So try from today. Just one nicotine, two E's are there, from nicotine to nicotinic acid. IC you have to add. Just try. But you have to add it biologically. Then you smoke cigarette and you prevent pellagra. Anyway, pellagra never happens nowadays. It's obsolete. Pyridoxal phosphate, it transfers amino group. All transaminase reaction require PLP. All decarboxylation reaction require PLP. The olden name of PLP is PALPO, PALPO. Olden names, very old names when it was first discovered. And this name was given by Bengali scientists. That time Bengal was good in science, not now. That time there was very big scientist. Meghna Shah, Satyanath Dotto, Satyanath ki? S.N. Bose. And now Sandeep Ghosh. So how the Bengal will prosper? I am very big scientist, only by size, not by merit. So this pyridoxal phosphate is nothing but the coenzyme form of vitamin B6. What is next? Why I am taking so much time for this slide? Because it is important for exam. And I tell you, in the final, these questions only will be asked to you and you won't be able to answer. Tetrahydrofolate. They transfer 1 carbon moieties or 1 carbon units. They are coenzyme forms of folic acid. Folic acid or folate. Thiamine pyrophosphate, TPP. Thiamine pyrophosphate or thiamine pyrophosphate. Thymine and thiamine, two things are different. Thymine found in DNA. Thiamine found in all cells. It's a part of vitamin. Thiamine pyrophosphate is a coenzyme form. The vitamin is vitamin B1 or thiamine. Correct? What does it do? It transfers aldehyde groups, carries aldehyde groups. There are two reactions which require TPP as coenzyme in our body. One is transketolase, another is pyruvate dehydrogenase. The same type of reaction is also catalyzed, same type of reaction catalyzed by another enzyme called alpha ketoglutarate dehydrogenase. So these two are in one type, another is transketolase. In both the cases, they transfer aldehyde group from one part to another part. Now, whatever I spoke about the structure, now it's in a flow chart. Let's see, enzymes, it might be simply only protein in nature. It will be a complex, protein plus non-protein part. Then apoenzyme and cofactor. Coenzyme is organic part, inorganic part, tightly bound, inorganic part, tightly bound, prosthetic group, inorganic and tightly bound. They are called prosthetic group. Correct? Now, how enzymes act? What is the mechanism of enzyme action? For every substrate, we require a separate enzyme, or some enzymes can react with many types of substrate. Which one is correct? To answer this question, first, if you understand the chronology, then you get this one coming first, then this one, correct? And this is latest, correct? In 90s, this came in 90s. But actually, this we read first because it's by the name of Emil Fischer. Fischer's lock and key model. Lock and key model. What do you see in the diagram? Let's check your observation power. Tell me, what do you understand about enzyme and substrate from this diagram only? একজন দাঁড়িয়ে বলো দাঁড়িয়ে বলবি, আমি তোর বাড়ির টিউশন মাস্টার নই যে তোর মা একটু পরে আমাকে চাউমিন খাওয়াবে। বলো, জোরে বলো। সেটা কি এখান থেকে বোঝা যাচ্ছে? আমি কি বল, ছবি থেকে বলতে বলেছি। খাপে বসে আছে, ভেরি গুড। তাহলে কি আমি কি বলবো, ইংরেজিতে কি বলবো? কমপ্লিমেন্টারি, স্ট্রাকচারাল কমপ্লিমেন্টারিটি, তাই তো? আর কিছু? Enzymes? Quantity is not the right word. Size, enzyme বড়, substrate ছোট. It is distinctly visible in this diagram. In 99.99 cases, enzymes are bigger, much bigger than the substrate, except the enzymes which act on nucleic acid, DNA polymerase, RNA polymerase, এই এনজাইমগুলো সাবস্ট্রেট থেকে ছোট হয়। These enzymes are only smaller than substrate. All are bigger than the substrate, right? Third point, comparatively a small region of the enzyme is responsible for interaction with the substrate. See, this whole area is left untouched by the substrate. Only a small part gets attached with the substrate. So 3 points we get. What is that? 1, the size does matter. Enzymes are bigger. Number 2, they have structural complementarity. Number 3, a very small region of the enzyme is responsible for binding with the substrate, which is called active site. Correct? This was given by Emil Fischer. Okay, lock and key theory. Now, what this theory tells actually? When I say lock and key theory, what comes to your mind when I say lock and key? Key fits in the lock, very good. Anything else? Every lock has its own key, every key has its own lock, very good. Anything else? Very good, that is the same answer. Have you ever seen a lock made up of mud, wood, soft toy? What is that word? Rigidity. Means both lock and key are rigid. Neither of them can change their shape. The mother-in-law wants you to put the veil up to here and you don't want to put on sari at all. Well, something else will put. What will happen? They cannot stay together. Fine. That means they are so rigid, neither enzyme will change its shape nor according to Emil Fischer. Fine. But then the problem came. What is the problem? In the first class I told you the problem. What was the problem? Different types of meat. The proteolytic enzyme for different meat should not be different. Enzymes produced by inside Hindu and Christian or Muslim, are they separate? Don't say yes. No. Whether you eat pork or beef or don't eat, or you don't take any meat at all, the enzymes will remain same. If someday by choice or by force you are made to eat or you like to eat the meat which is forbidden according to your religion, nothing will happen. You won't have any medical problem. Yes, ethical problem might be there. Oh my God, what I have done. With this feeling you might get diarrhea, but not due to the food. When you eat something with a very distasteful manner, that food you cannot digest because of the anxiety. So you can digest. So the question came, then is it possible to prepare so many enzymes for so many different types of meat? Shrimps, prawns, large prawns, big prawns, medium prawns, crabs, king crab, small crab. What is this? Wall lizard, monitor, water lizard, কি ড্রাগন আইল্যান্ডে পাওয়া যায় ওই কোমোডো ড্রাগন, then out of crocodile, see caiman, gharial, saltwater crocodile, Florida crocodile, so many things are there. Rock python, snakes if you come. Is it possible? No. So then this man Koshland came up with this induced fit. What is an induced fit? Means you come to an adjustment with your mother-in-law. Okay, I will wear sari, but the veil will be up to here. Okay for you? One says, okay, fine. That means you also change somewhat, she will also change somewhat. And then we both can stay together. It's called induced fit. But here, both is not changing. Enzyme is changing with respect to substrate. Whatever may be the substrate size, shape, enzyme will somewhat manage with that altered shape. Active site is flexible, not rigid. That's why no lock and key. The moment this theory came, then the answer for digestive enzymes fallacy was solved. Now, these people, Wolfenden and co-workers, they came up with this theory, that is preferential transition state binding theory. Now, this says, first one was neither enzyme nor substrate will change their interface. Second one, enzyme will somewhat malleable, and the subtle changes will be managed by the enzyme. This one says that when what has changed here? Both have changed. And they found, how they found? They took, they freeze dried, means lower the temperature, got this reaction frozen at one point. একটা particular point তাকে freeze করে তার একটা molecular ছবি তুললো, ঠিক আছে, একে বলে এক্স-রে ক্রিস্টালোগ্রাফি। তার ছবি তোলা হলো, ছবি তোলার পরে the photograph revealed this one. Now this shape is neither like this, the original native state of the substrate, nor it holds the original native state of the enzyme. It is something new, something completely new. That means the enzyme and substrate both change their shape in order to bind more tightly. In order to bind more tightly. Correct? How to prove that this theory is correct, not the other one? They designed two sets of inhibitor. One inhibitor which is similar to this shape, another inhibitor similar to this shape. Two inhibitors, one similar to the actual shape of the or native shape, native structure of the substrate, another the shape of the substrate condition bound with the enzyme. This particular state is called, can you tell me what is this? Yes? Transition state. So the enzyme and substrate bind with each other more tightly in their transition state, neither their native state structure of the enzyme nor the native structure of the substrate. So they found when they prepared the inhibitor similar to this is more active compared to the native one, and that's how they proved